My name is Mark Harris. I'm a paediatric endocrinologist at the Lady Cilento Children's Hospital. The thing about type 1 diabetes is it's often diagnosed early in life, so usually during the first decade, and it's a lifelong condition. Unfortunately, there's no cure, and it has a significant effect on the family and the child, and as a result there's a very high cost to both the family and society. One of the things about type 1 diabetes is that although there's a family risk, 85% of children who are diagnosed with type 1 diabetes don't have a family member with diabetes. And in addition, although there are genes which increase your risk of developing diabetes, they're not that good at identifying an individual that will go on and get diabetes. The other thing to be aware of is that although it's common, it's becoming more common. So the disease rate is probably doubling every 2 decades since the middle of last century. So it just highlights the fact that we don't really have a good way of identifying which children go on and develop diabetes. And it highlights the fact that there's a gene-environment interaction. So one of the things, or the main aim of this study, is to try and identify those children who will progress to developing antibodies through looking at their gene activation pattern early in life. So the antibodies are very important because they're probably one of the best ways that we can identify progression to type 1 diabetes. So that was the essential aim of this study, to try and highlight those patients that are going to go on and develop antibodies. We see 2 to 3 new patients a week, and probably 70 to 80% of all the patients seen by the endocrinology department would have type 1 diabetes. The advantage of monitoring someone who's at risk of developing diabetes is that they're less likely to present with diabetic ketoacidosis, which is a medical emergency. So to put that into context, in children that are part of a monitoring program, the incidence of diabetic ketoacidosis at presentation is less than 5%. Currently in our hospital we're seeing rates of up to 35 to 40% of children presenting with diabetic ketoacidosis. So that's not a good start to managing their disease. By population screening we'd be able to reduce that dramatically, which would be a fantastic thing to do.
I am Dr. Ahmed Mehdi. I'm working with Professor Anjani Thomas as a postdoc. So in this research we used all the available data, available gene expression data available in the world, and specifically we are looking into longitudinal gene expression data, and this data basically are from 2 cohorts. The first one is the German baby diet cohort, in which we have access to the data from 109 children genetically predisposed to type 1 diabetes, and there are 109 subjects with 454 microarray data points measured at different times, and this is the number of groups which are seroconverted, and this represent a group which are non-seroconverters, and this represents the number of data points in each group. The second cohort is the Finnish DIP cohort, and we have access to 60 children. In this research we propose a sophisticated way of correcting the time, in which relative to clinical anchor in our research, the clinical anchor is the development of type 1 diabetes or to antibodies. For example, you can see in the slide that this is the subject 1 that is measured, and the green dots represent different time points of measurement peripheral blood, and the black dot circle represent the time at which this child develops autoantibodies. Similarly, this is the profile of the second subject, and this black circle represent the development of autoantibodies in this subject, and similarly we have a number of different subjects with different profiles and different time of development of type 1 autoantibodies. And you can see from here the data is heterogeneous and there is noise in the data. We have corrected the data using the methodology presented in this paper, and for example we use the time of seroconversion as an anchor or reference point, and this is a time point before seroconversion, this is a time point after seroconversion, and then we performed differential gene expression analysis on this data, and we found a filtered list of 67 genes. So we got 67 genes in these children that are differentially expressed as shown in this diagram. On x-axis you can see the time relative to seroconversion. This time 0 represents the time when the subject get seroconversion, and this is the time before seroconversion. And similarly, since antibody negative subjects do not seroconvert, so we corrected relative to mean seroconversions shown on x-axis. The colors represent the red is the genes that are highly expressed, the blue are which are low expressed, and the time shown on x-axis, and each row represents the gene expression profile at different time intervals. So if you look into this diagram, you can see a group of genes that are highly expressed at near birth, and this is another group of genes that are not highly expressed, they are colored in blue and at near birth. And this group of genes have a similar profile as that of non-seroconverters, and some genes didn't change throughout the time, and they were different in the both groups. So this was very interesting to see a different patterns of gene expression, and you can see in this pattern is not as clear in seroconverters, so there is a disruption of this pattern in seroconverter. Our aim was to predict the subjects who are likely to seroconvert. We used some mathematical approaches to get a filter list of 7 genes and an HLS score, and we used a prediction model, which is a logistic regression model, and that predicts seroconversion by taking a data at or near birth. So you can see in this the diagram. This is the ROC curve, which represents how accurate the model is. On x-axis it is the specificity, on y-axis it is the sensitivity, and the area under the ROC curve is 0.85, which is very high, which represents the accuracy of this model. If we use this model to predict who are going to be not seroconvert, you can see from this line that if the model predicts to be non-seroconverted, the subjects remain seronegative for 90% of the time. So this represents a very high clinical value of this model. In conclusion, so we have presented a novel method of data correction where we correct relative to seroconversion, and when we performed differential gene expression analysis, we found 67 genes that are differentially expressed, and we have seen a disruption of gene expression profiles in seroconverters. And when we use 7 filter list of 7 genes and HLS score, we predict subjects who are likely to seroconvert and subjects who are not likely to seroconvert with the accuracy AUC of 0.85 and the sensitivity and specificity above 80%. So next steps are we are looking to validate our findings through some independent cohort, and some gene expression data is expected to release by the end of this year that we'll be using to validate our findings. And we are aiming for population screening way to use this model to see who are going to seroconvert in a normal general population who are at high risk of developing type 1 diabetes.
I'm Ranjini Thomas. I'm Professor of Rheumatology at University of Queensland. In the future, we can follow cohorts of children at very high risk and institute prevention measures, including trials of things that we can do to prevent the disease in the future. The second important thing about this study is that because we've looked at gene expression early in life, our study gives a very important underpinning understanding of how the immune system is involved in the development of type 1 diabetes early in life, well before the development of antibodies and before the actual development of type 1 diabetes.